All living things breathe air all the time. This apparatus shows the difference between the inhaled air and the exhaled air. We see the air in this room is composed of 21.15% oxygen and 0.07% of carbon dioxide. When the athlete exhales into the apparatus, we see it's composed of 16.11% oxygen and 4.10% of carbon dioxide. This demonstrates that the exhaled air contains less oxygen and more carbon dioxide. In other words, when we breathe, we use up oxygen and give out carbon dioxide. Place a rat into a sealed plastic box connected to an air pump. Turn on the pump to draw air into a flask containing sodium hydroxide solution to remove the carbon dioxide in the air. The air is then drawn into another flask containing red hydrogen carbonate indicator. The color of the indicator changes according to the percentage of carbon dioxide in the air. Air containing 0.03% of carbon dioxide turns the indicator red. With more carbon dioxide turns the indicator yellow. Air without carbon dioxide turns the indicator purple. In this experiment, the indicator changes to purple slowly, showing that there is no carbon dioxide in the air. We then pass this carbon dioxide-free air into the plastic box for the rat to breathe. After that, air is directed to pass through the red hydrogen carbonate indicator again. The result shows that the indicator has turned yellow, indicating the existence of a large amount of carbon dioxide, which has been exhaled by the rat. This experiment shows that carbon dioxide is released when an animal breathes. When we blow air onto a mirror, it becomes misty. A piece of blue cobalt chloride paper placed onto the misty area changes to pink, indicating the substance on the misty area is water. In other words, exhaled air contains water. Living things breathe so as to take in oxygen and give out carbon dioxide in return. Why does this exchange of gases occur? Place a rat into a thermos flask. Keep another thermos flask empty as a control. Connect two test tubes onto the two ends of a U-tube containing red ink. Place the test tubes into the two thermos flasks respectively and seal the openings with cotton balls. The watermark inside both tubes lies at the same level, indicating equal air pressure on both sides. 2 hours later, the watermark of the test tube in the empty thermos flask rises while the other one lowers. Why? It's because air pressure of the test tube in the thermos flask with the rat has increased. This is probably due to a rise in temperature in the flask, causing air in the test tube to expand. This indicates that heat energy is released when the rat breathes. Breathing brings oxygen into the body of a living thing. Oxygen is eventually brought into the cells. chemical reactions between oxygen and the food stored in the cells occur, releasing energy and carbon dioxide. The energy is immediately used in metabolism. This process is called respiration. Place a piece of butter on an evaporating dish and heat it until the butter catches fire. The flame raises the temperature of a small amount of water. This demonstrates that burning food releases energy the same way as respiration. Place the burning butter beneath this apparatus to collect the gas released. Test the gas with lime water. The lime water turns milky. This indicates that carbon dioxide is released in the burning process. In the process of food burning, all the stored energy is released within a short period of time. On the other hand, respiration is a series of biochemical reactions, during which energy is released slowly and continuously for full use by the living thing. In other words, respiration is the process during which oxygen is used to oxidize food, while energy, carbon dioxide and water are released. We can design similar experiments to study respiration in plants. Place a living green plant inside a sealed plastic box and cover it with a black box to stop photosynthesis. Turn on the air pump to let air pass via the tube into sodium hydroxide solution for removal of carbon dioxide. The air is then passed into a flask of hydrogen carbonate indicator. The indicator turns purple, showing that the air is carbon dioxide free. Pass the air into the box containing the green plant. After a while, the hydrogen carbonate indicator in the flask on the right turns yellow, indicating the existence of a large amount of carbon dioxide in the air passed out of the box. This experiment shows that when plants respire, carbon dioxide is released. Pour some alcohol onto a funnel containing germinating seeds to kill the germs on the surface. Then place the seeds into a test tube and put in some cotton balls holding sodium hydroxide pellets. Insert a rubber stopper with a capillary tube. Seal with adhesive tape. Add a drop of red ink into the capillary tube. Place the test tube into a beaker of water at a constant temperature. Prepare an identical setup containing boiled seeds as the control. The test tubes in both setups are kept at the same temperature. Record the level of the red ink in both capillary tubes. After a while, the red ink in the setup with germinating seeds moves downward, indicating a decrease in the amount of air inside. Meanwhile, the ink drop in the control shows no change in position. Why is this so? Germinating seeds are alive. They undergo respiration, during which oxygen in the test tube is consumed and carbon dioxide is released. But the released carbon dioxide is immediately absorbed by the sodium hydroxide, resulting in a decrease in the volume of air inside the test tube. Therefore, the red ink moves downward. Meanwhile, the boiled seeds in the control are dead. They cannot undergo respiration, and the volume of air inside the test tube remains unchanged. Hence, the red ink does not move. The results in the two setups indicate that plants consumed oxygen during respiration. Fill a thermos flask with sterilized seeds. Insert a thermometer into the mass of seeds and fix it with a cotton ball. Seal the thermos flask and place it upside down on a frame. Record the initial temperature. Prepare a control using seeds killed by boiling. Record the initial temperature. After a day or two, there's a slight change in the temperature inside the control due to the change in room temperature. But in the setup with sterilized seeds, there's a marked increase of temperature. The results indicate that when plants respire, heat energy is released. It seems that plants do not need food to stay alive. Why is this so? In fact, plants produce food for living by combining carbon dioxide with water. Light is necessary for this process, which is therefore named photosynthesis. The green waterweed produces air bubbles under illumination. Collect the bubbles in a test tube and insert a glowing splint into it. The splint relights, showing that the gas released by the waterweed is oxygen. Place a green plant in a dark chamber for several days so as to eliminate the product of photosynthesis in the leaves. Take out the plant. Cover part of a leaf with a piece of aluminium foil. Leave the plant under light for several hours to carry out photosynthesis. Then cut off and test the leaf. Place the leaf in hot water to kill its cells and to allow chemicals to pass through the cell membrane. Next, transfer the leaf into heated alcohol to remove the chlorophyll. The leaf is bleached so as to better reveal its structure. Place the leaf into warm water to remove the alcohol inside the leaf cells. Finally, place the leaf on a piece of white tile. Add drops of iodine solution onto the leaf. The uncovered part turns dark blue, while the covered part shows the color of the iodine solution. The principle of the iodine test is that when iodine mixes with starch, the mixture turns dark blue. In other words, any material which turns dark blue in contact with iodine solution contains starch. This experiment shows that green leaves produce starch during photosynthesis. Based on the experiment, we can conclude that when a green plant undergoes photosynthesis, chlorophyll in the leaf absorbs the energy of sunlight. The energy is passed to the absorbed water, which releases oxygen and reacts with carbon dioxide. Starch is eventually produced and stored in the plant. In nature, living things get their food from other animals or plants. For example, vegetables in our meals come from plants, whereas beef and other meat come from animals. However, cows feed on grass. Hence, people who eat beef indirectly rely on plants. This example shows that while a cow eats grass, a man eats beef, which is the flesh of a cow. The energy fixed by grass through photosynthesis is passed to the cow. The energy stored in the cow is eventually passed to the man. This pathway of energy transfer is called the food chain. As plants can produce food for themselves through photosynthesis, they are the producers in the food chain. Animals, on the other hand, cannot produce food for themselves and rely on plants for food directly or indirectly. They are consumers in the food chain. All food chains in nature begin with plants which are capable of undergoing photosynthesis. Hence, photosynthesis is the most important process in nature. Animals breathe all the time, taking in the oxygen in the air and releasing carbon dioxide. Plants also respire all the time, consuming the oxygen in air and releasing carbon dioxide. Burning also consumes oxygen and releases carbon dioxide. Will there be more and more carbon dioxide but less and less oxygen in the air? No, this will not happen. The reason is that green plants under sunlight undergo photosynthesis while respiring. Hence, they consume the carbon dioxide in air and release oxygen in return. We all agree that green plants are very important to the earth and mankind. The reason is that green plants can undergo photosynthesis, which helps maintain the equilibrium of oxygen and carbon dioxide in the air.